SF3B1 mutation occurs in a very high frequency in CLL. This gene mutation has a very high co-occurrence with ATM mutation as well as 11q deletion, for which the minimal deleted region contains ATM. So suggesting that there is a synergy between these genetic lesions. Our previous data have already demonstrated that expression of heterozygous SF3B1 mutation in murine B cells doesn't lead to CLL alone. Instead, co-expression of ATM deletion with SF3B1 mutation can co-drive the onset of CLL, highly suggesting that these two genetic lesions contribute together to the leukemogenesis. To try to understand the molecular mechanism underlying these two genetic lesions, we actually focus on one molecular mechanism called centromeric R-loop formation. This is mainly because centromeric R-loop has been previously demonstrated to be important for chromosome segregation. So we first examined the centromeric R-loop level in SF3B1 mutant cells using immunofluorescence staining. Our results clearly indicated that SF3B1 mutant cells has very high level of centromeric R-loop formation. We then carry for the consequence of the increasing level of centromeric R-loop. Our result indicating that this increased level of centromeric R-loop actually can lead to mitotic stress with increasing level of misaligned chromosomes. So we therefore examine how this increased level of centromeric R-loop can lead to mitotic stress. So we have been focused on the chromosome oscillation as well as a spindle defect. Our result clearly indicating that if we have SF3B1 mutation, these cells has increasing level of hyperchromosome oscillation as well as a spindle defect, which eventually leads to increased level of aberrant mitosis. So, and in the situation upon the both SF3B1 mutation and ATM deletion, so the defect is even more drastic. We further search for the potential contribution of splicing factor mutation involved the specific variants into this centromeric R-loop formation. We are able to identify some of the key RNA binding protein which can involve in the biogenesis of R-loop. So we hope with that we can close the story on the splicing factor mutation, specific variants, as well as chromosome instability.